So in part 5 of chapter 4, we'll take a look at molecular phylogeny. Molecular phylogeny has been used more frequently in recent years as the price of DNA sequencing and the analytical tools that we have to analyze trees has become more widely available. And it's been used in many cases to resolve phylogenies that we have not been able to resolve with other means. And one good example of that is the origin of whales and whale phylogeny. We know that whales belong somewhere in this particular grouping, but the big question has always been, do whales belong within this group, what we call the artiodactyls, which are also referred to as the even-toed ungulates. So these are even-toed ungulates here, all of them are, and this is that ancestor that leads to the artiodactyls. We see the two dotted lines here. So one of the hypotheses is that the whales are actually inside that lineage. So they're inside the artiodactyl group if I look at this first position of the whale here. If I look down here, this other dotted line, I can see that this places the whales outside of the artiodactyl group. So it places them as a relative to the artiodactyls, but not within the artiodactyls. And so that has been a question for many years, where we should be placing the whales on this particular tree. Now, if we look at just parsimony analysis of placing whales as the closest kin to artiodactyls, so in this particular case, parsimony analysis on various phylogenetic, parsimony analysis on many morphological characters place the whales here. So it places them outside the artiodactyl group, not within the artiodactyl group. However, that placement is under question because that whole grouping that we saw, the artiodactyl grouping, is based and hinges on a very important bone structure. It's a bone that is found in the ankle. It's called the astragalus, and it's the shape of the astragalus. So we see two different ones here. We see the artiodactyl condition, and then we see the non-artiodactyl condition. That split in the ungulates between the non-artiodactyls and the artiodactyls is based upon the shape of that particular bone. Well, how do we assess the structure of that bone in an animal like a whale that doesn't have the bone? That bone has been lost. So if we've lost the ankle, we've lost that limb, we can no longer assess that morphological trait. That is why we're turning now to molecular phylogeny to try to understand where to place the whales in that particular phylogenetic tree. Here is a video from Oxford University Press that talks about creating phylogenetic trees using sequence data.
Chapter 4. Building phylogenetic trees. A phylogenetic tree is a diagram showing inferred evolutionary relationships between a set of organisms. Phylogenetic trees have a branching pattern, like this figure from Darwin's notebook, where he has sketched out this branching pattern to reflect the process of descent with modification, a process central to Darwin's theories of evolution. This second tree shows the inferred evolutionary relationships among Darwin's finches on the Galapagos Islands. But how are phylogenetic trees like this one built? Well, any characteristic can be used to infer relationships and build phylogenetic trees, and DNA sequence data has proven incredibly helpful in clarifying and building many important phylogenetic trees. Using large DNA datasets to build trees can get quite complicated, but the underlying principle and logic is pretty straightforward. Sequences separated by shorter evolutionary distance are expected to be more similar to one another than sequences separated over longer evolutionary distances. We'll go through a highly simplified example to familiarize ourselves with the basic logic of phylogenetic tree building from DNA sequence data. We will do this in a series of steps so that you can pause the video at any point to work through the steps yourself, returning to the video to check your progress. In our simplified example, we have a set of 6 sequences. These are the equivalent stretch of sequence taken from the equivalent gene from each of the 6 species, and we're just going to refer to the species as A through F. The first step in the process is to line the sequences up against one another. Here we have the sequences aligned so that the equivalent nucleotide in each sequence lines up to form a column. This makes comparing the sequences much easier. With the sequences aligned, we can now compare each sequence with one another. We'll do this in a pairwise fashion, starting with sequences A and B. So our next step is to determine how many differences there are between sequence A and sequence B. When we do this, we see that in 3 positions the sequences are the same, and in 9 positions they are different. So we have 9 differences between sequences A and B. We can start building a table of all the differences between the sequences, and our first entry is 9 differences between A and B. The next step is to compare sequences A and C to determine how many differences there are between these 2 sequences. In this case, we can see that the sequences are much more alike, with 10 positions that are the same between the sequences, and only 2 that are different. So our next entry in the table is 2 differences between A and C. And we can continue comparing the sequences in this way until we have completed the rest of the table. This is our next step. And this is what the completed table looks like. With the table completed, we can move to the next step, which is to use the table to identify the sequences with the fewest differences between them. We will infer that these are the sequences that are the most closely related to one another. In our table, we see that the sequences with the fewest differences between them are A and C, with only 2 differences, as well as B and E that also only have 2 differences between them. With this information, we can draw the first groupings on our phylogenetic tree. We'll group A and C together to reflect the fact that these 2 sequences show the closest relationship we have here to one another, and we'll group B and E together to reflect the fact that they also show an equivalently close relationship to one another. With the first 2 groupings made on our tree, we now need to rework our table, with the grouped sequences combined together as a grouping rather than 2 individual entries in the table. We'll start by combining A and C, this group. To do this, we'll take the average difference that A and C show to each of the other sequences. Let's start with the differences they show to B. We see there are 9 differences between A and B, and 9 differences between C and B. So the average difference of A and C to B is 9, and we can make that entry on our new table. Let's move to the next position, D. We can see there are 4 differences between A and D, and there are 5 differences between C and D. So the average difference of A and C to D is 4.5, and we can add that entry on our new table. We can complete the rest of the table in this way. We have an average of 9 differences between A and C to E, and 10 differences to F. The rest of the table can be copied down from the first table. With the AC grouping now added to our new table, we can proceed to also add the BE grouping. And to complete the table with B and E grouped together using the same approach as before. For B and E, we have an average of 9 differences to the AC group. We have 4.5 for AC to D and 10 for AC to F. For B and E to D, we have an average of 6 differences and an average of 10 for B and E to F. And there are also 10 differences between D and F. With this table completed, we can now proceed to the next step. This step is to identify the sequences with the fewest differences between them in our new table. We can see that the AC group has only 4.5 differences to D, so this is the next close relationship in our tree. And we can add this next grouping to our tree, like this, with D as the next grouping out from A and C, reflecting the fact that D is more closely related to them than it is to the other sequences we have. With this new grouping added to the tree, we need to rework our table again, with the ACD grouping incorporated. We have an average of 7.5 for AC and D to the BE group, and we have an average of 10 for AC and D to F. And we also have 10 differences between the BE group and F. With the new table completed, we can now determine the next relationship by identifying the sequences in the table with the fewest differences between them again. We can see that this is the ACD group with the BE group, with an average of 7.5 differences. So we can add the next grouping to the tree, like this, grouping the ACD group with the BE group. This now leaves us with one more sequence, F, that is equally distantly related to all the other sequences, with 10 differences to each of them. So we can add this last grouping to the tree like this, reflecting the distant relationship between F and all the other sequences. And this completes our phylogenetic tree built from these 6 DNA sequences. The method we use to build this tree is a distance method, specifically an approach called unweighted pair group method with arithmetic mean, or UPGMA. And the approach we took works well for a straightforward situation like this, but when we start to look at larger and more complex data sets of DNA sequences, things can become more complicated, and we need to start taking a number of other factors into account. And it's often a good idea to compare the results from multiple different approaches to develop a sense of how reliable different parts of your tree are. But despite these potential complications, the underlying logic remains that sequences separated by shorter evolutionary distance will be more similar to one another than sequences separated over longer evolutionary distances. DNA sequences therefore provide a powerful source of information to help us infer evolutionary relationships that can be depicted in the form of phylogenetic trees.
Here are some sequence data, a portion of a gene that is used for making beta-casein. We align these up. We have our outgroup down here, and so the outgroup is our comparison. That represents the ancestral condition. So the ancestor had a G there, and if we look at position 142, everybody in the group has a G there as well. These are all homologous spaces right here, but they don't tell us anything. Remember, we looked at the idea of non-informative characters in our video when we talked about the antelope, and we said sometimes there's characters that just don't tell us anything. If everybody in the group has the same character, then it doesn't tell us anything, because remember, what we're trying to do is to group them based on shared derived features. So if I can find something that's different from the ancestor here, but that a group of them shares, then that might be an evolutionary event that occurred leading to that particular branch. But if they're all the same, then I can't do that. So position 142 is not telling me anything. And of course, any of the ones along here where they're the same, they also are not really telling me anything. So they come over here to position 162, and position 162 gives us a little bit of insight, because what we see is that the ancestral condition is a C, and the camels, peccaries, and pigs all share that same ancestral condition. So that would suggest perhaps that they are closer to the ancestral state. And then the 4 species up here, the cow, deer, whale, and hippo, they all form their own cluster. So they have a shared derived feature by having a T in this 162 position, suggesting that they go together on that tree. Likewise, if we come over here to position 166, we see that the ancestor had a T, and we see that these two here, the whale and the hippo, have something different from the ancestor. So they have a shared derived feature, and that is the C. So it could be that the ancestor leading to this lineage that gave rise to both whales and hippos had an evolutionary event there where a base became C, and then the cow and deer perhaps had a similar situation where they had that position become a G. So that is telling us, that is again kind of suggesting that the whales and hippos go together. Over here we have a C in the outgroup, so that's the ancestral condition. The cow and the deer are closer to the ancestral condition, they share it. But then if we look here, the whale, hippo, pig, and peccary, they actually share a derived trait because they all have the T. It's also mentioning that 192 is also not an informative character. So 142 and 192 are not telling us much, but 162, 166, and 167 perhaps are. Looking at lots of different trees for many other characters than we see here, we look at these 2 different possible trees, and so they're only putting the traits that we looked at here on this tree. And remember, we had really 2 major questions we were asking. Do the whales go in here next to the hippos, or do the whales come out here outside the entire artiodactyl group? And so if we go through and we create the tree based on, again, some of these other characteristics, and ask about the different changes that would have to occur. So if this is the way that the tree is going to look, if this is how I'm going to place them, and I'm going to place pig and peccary close together, I'm going to put the whales and hippos close together, and then I'm going to put the cow and deer next to one another as sister groups. If that's what it looks like, then I can go through and ask what these evolutionary changes were. So back here we could see that in position 166, a T became a G. In this lineage that led to the camels, we have the T became an A. Okay, so that would explain what happened at least initially with position 166, but then this lineage that led to the whales and hippos, what had become a G now becomes a C. So the G becomes a C. And then as far as position 162, we see that the top four here have the same base. So in that case, this is where the C became a T. And we also see down here for position 177, it looks like a C became a T in this particular group. So if this is the way that it happened, then we go and we count up how many different changes would be necessary to get this type of a tree, and in this case we get 6 changes. If we do the same thing, but instead we put the whales out here on the outside of the artiodactyls, but still we end up grouping the pig and peccary together here and the cow and deer together up here, we do the same sort of thing and then ask what kinds of changes would be necessary. We end up needing 9 different changes. So it would appear that the most parsimonious solution would be this one on the far left, where the sister group to whales is the hippos. So again, this would suggest that these are more closely related to one another than they are to anyone else on this tree. Now, we said before that searching for possible trees and then the best of the possible trees can be very difficult. It can be very labor intensive because, as it shows here, the more species you add onto your tree, the more possible trees there are. In the book, they talk a little bit about some of the different things that they do to try to resolve some of this and to search for these. We aren't really going to go over these. Your textbook goes into some of this in some detail about how we would go about looking for the correct trees, and we're omitting that part of it. Now, one of the techniques that is often used in trying to determine these trees and to draw them is what's known as a distance matrix. And then once you have a distance matrix, you can use this technique called neighbor joining. So basically what you would have is you would have a DNA sequence and you would look at the amount of similarity between the sequences. And where this number is very low, it means that there are very few differences. So this distance matrix is actually looking at how different they are. This position right here is looking at whales and hippos. So the differences we see in the sequences between whales and hippos gives us a value of 0.053. So if these were identical, then the distance would be 0. So if we had a whale-whale comparison here, which we don't do, but if we did that, that would be a 0. And so if they were exactly the same, there would be a 0. And so you can see one of the very low numbers we have in here is this 0.053, the distance between whales and hippos. We also see a low number here between deer. We see a 0.07. We see a fairly high number between the outgroup and the deer. We see a pretty high number between the outgroup and the cow. So what they do is they start off with what they call here the star phylogeny. Here's your outgroup and here's everybody on it. And then they've looked at some of the lowest numbers that we see here, the cow and the deer. And so they're going to put the cow and the deer on a branch together. And then if we go down and we look at the hippo and whale together on a branch because they have a very low number. And we can put pigs and peccaries on a branch together because if we come here and look at pig and peccary, they also have a low number. They essentially put them into these pairwise distances and put those on a tree, and then also try to look at the difference between this pair of species and that pair of species. So we might look at the difference, say, between a hippo and a deer to try to figure out what this distance might be, and we can do that here. So this is what we might refer to as an unrooted tree because we don't have any true ancestor to it. And then over here we root the tree and we base it on this. So the camel was the first one to come off of the outgroup. Then we see the pig and the peccary here next to each other, the whale and hippo next to each other, and then the cow and the deer next to each other. Another molecular technique we can use are signs and lines. Signs and lines stand for short and long interspersed nuclear elements. So these are sections along the DNA that are typically interspersed between those sections that are actually coding for proteins. And what will happen is these repeated sequences get laid down between the genes, and the positions of where they're laid down is very distinct. And so two organisms that have signs and lines in exactly the same locations share a common ancestry. And then if they have different signs and lines, they are less related to one another. So the more signs and lines they have in common, the more closely related. So what they do here is they're looking now, going along sort of the position. Locus is a position along the DNA molecule. So let's look at a particular position along the chromosome, and where there is a 1 there, they share that particular sign or line. Where it's a question mark, then we don't know whether they share that or not. If we go in here and we look at whales and hippos, we can see that whales and hippos share signs and lines at positions 4, 5, 6, and 7, and they share that with one another, and no one else in this group shares that with them. So they are unique in sharing that 4, 5, 6, and 7, which is why they also put that 4, 5, 6, and 7 here. So this sign or line was laid down in the DNA, in the lineage that led to the whales and hippos. Now we know that whales and hippos then also have slight differences, because whales have a sign or line here in position 1, 2, and 3 that hippos don't have. So that came after they split off. If we look over here, let's say between cow and deer, we can see a lot of similarities here between, we have position 8, 10, 11, 12, 13, 14, etc., 15, and 17. So what they're showing here is these positions, the cow and the deer all have those. Now the reason why they put 9, 10, 12, and 13 outside of that is because they're shared not only between cow and deer, but also with the whales and the hippos. So if I come here and look at position 9, they're suggesting that these 4 share that trait. So the cow, deer, whale, and hippo all share the one at position 9. They definitely share the one there at position 10. They share the one at 12. They share the one at 13. So all 4 of those shared these signs and lines. So that's how they're building those. And again, we can go through and look at similarities and changes in those for the presence or absence of that sign or line in that position. So we don't use the absence as any sort of indicator. The zeros don't tell us anything, but the presence of the sign or line in the same place does tell us something about ancestry. So this supports what we saw before with the beta-casein gene, is that it looks like the hippos come out to be the closest living relatives to whales. Now, new fossil evidence that have been found suggests that this is in fact correct, because more recently we have discovered a fossil of a whale that looks like a transitional species, showing us changes that occurred back when they still had their ankle bones. So when we go through and we look at ancestors that still had that astragalus bone, it does appear to look more like that we see in the other artiodactyls. Therefore, they probably should go within that group and not beside the group. These videos I will post in my courses. One is looking at a website that allows you to look at an interactive phylogenetic tree tutorial. The other one is on creating phylogenetic trees in lizard populations.